Hello and welcome to our Odin Analysis Spotlight video series. Today, we are going to take a look at how you can leverage the Odin Community Analysis software package to learn more about microbial communities like those found in soil using Shannon's Diversity Index, Shannon's Evenness Index, Average Well Color Development, and EcoPlates.
EcoPlates are a great drop-in solution for assessing the flexibility of a microbe or community of microbes with regard to what carbon sources can be utilized for energy production. The EcoPlate contains 31 unique carbon substrates selected to probe various pathways known to be important to environmental isolates. Odin then takes care of the incubation and the reading, while the software takes care of the analysis. When comparing communities and their metabolic flexibility, it's important to take a look at a variety of factors like overall energy output, diversity of substrates utilized, and how effectively substrates are used relative to one another. With Odin Community Analysis, we use 3 different metrics to make this easy. Here's an example of the average well color development, which shows overall energy output, Shannon's diversity, which is related to the number of carbon substrates utilized, and Shannon's Evenness Index, which is related to how evenly those substrates are used. These curves enable you to draw conclusions about how many and how evenly carbon sources are utilized by a community and are easy to generate using the Odin software, where complex manual calculations may otherwise be necessary. So let's take a look now more deeply at the Odin software to learn more about the different metrics which can be helpful in learning more about your communities. Here we're going to be taking a look at 2 microbial communities from different soil samples. So first I'm going to pick out the community analysis project I want to analyze. And then for a reference sample, I'm going to pick CM1, which comes from soil rich in decaying organic matter. The test sample I'm going to compare to CM2 comes from dry compacted soil with little organic matter. I'll then select our plate type and hit generate charts to get things going. So taking a look first at average well color development, which, as the name implies, takes an average of the signal in all wells over time and enables you to get a bird's eye view of the overall energy output of your community. Unsurprisingly, CM1, shown here in black, shows much more robust energy output than CM2, shown in orange, which is consistent with having many microbes working actively to break down organic material. Shannon's Diversity Index here refers to the diversity of substrates which were utilized by the community. A high diversity index means that many substrates are being used. Comparing CM1 and CM2, we see that CM1 shows a rapid increase in diversity of substrates early on in incubation, whereas CM2 lags behind and does not metabolize the same variety of substrates, as indicated by the fact that it does not reach the same height of Shannon's Diversity Index. Noise caused by high background in this index can be controlled here using the pattern development cutoff, which is the total signal in all positive wells. Shannon's Evenness Index here is related to the rate at which substrates are utilized. As evenness approaches 1, substrate utilization has reached an equilibrium, where a spike can mean that only a handful of substrates are being used. Finally, CM1 shows a steep spike indicating uneven use of a few substrates before slowly coming down to 1 as more and more substrates are utilized. CM2, only using a few substrates, shows a faster drop to equilibrium. Noise caused by high background can be controlled here using the positive response cutoff, where only wells which reach that threshold of absorbance can be counted. With average well color development, Shannon's Diversity, and Shannon's Evenness indexes in the Odin Community Analysis Toolkit, comparing microbial communities' metabolic profiles is easy and informative.
Thank you very much for tuning in to this Odin Analysis Feature Spotlight. If you have any questions or are interested in trying this analysis yourself, please reach out to info@biolog.com. Or to find more Spotlight videos and other helpful information, please visit biolog.com.